by my own background, which is photonics and electronics. So even though my major is still in photonics or optoelectronics, I still had a chance to really involve in all these different fields. Therefore, just a very quick note, I personally think research process includes reading, experiment, publish, and networking. So you need to read papers as much as possible. You don't really need to study too detailed, but try to look as much paper and capture the most ideas as possible from other groups. For your experiment, you need to be very accurate and try to avoid making careless mistakes. One thing which I always tell my students is you have to plan ahead and always have your backup plans. So honestly speaking, during my PhD, I did not spend much time in the lab. I would say I spent the less time in the lab. However, I graduated also with the less time in the whole the school. The reason is I actually did plan a lot, and I did always know what I had to do during each experiment. So if you do have a very, very accurate and very successful planning each time when you do an experiment, you can actually save a lot of time during your PhD. So most PhD students, they actually do their experiments, and then eventually each week, 80% of data is garbage. So you only develop 20% of data to your PI. However, that's not very efficient. So planning is very important. And finally, you will publish. So publish step by step. You can expect to publish Nature and Science. However, you have to prepare the worst case. So it's especially for new PhD students, it's better to step by step and improving your publish. For a postdoc, I would say, of course, you need to be prepared and try to expect for the highest publication. And finally is networking. So networking is actually very important no matter you go to industry or if you go to academia. You need to make friends and you need to collaborate with a lot of people. Okay, so finally comes to today's main topic is biolaser. Okay, so here I will give an introduction of what is biolaser. So biolaser obviously has the phrase of laser. So laser is composed of 3 components. The first component is external energy source. So usually that is like this is a battery, or sometimes it's a laser pump. You need a mirror, and you need a gain media inside. So these are the 3 components of a laser. However, the definition of a biolaser is when you replace this gain material. So the traditional gain material is a piece of crystal, or a CO2, for example. If you replace this gain material with biologically related materials, so that could be your cells, your DNA, your blood, your tissue, whatever. So all by putting these biologically related material inside of the cavity or inside the 2 mirrors, when you pump it, you will expect to generate laser emission. So keep in mind that here we are not utilizing this laser, like what I'm using right now, as an illumination source. We are using this laser as a new type of detection method. So laser, the A stands for amplification, so light amplification. So which means anything changes inside here will be amplified. So by taking advantage of this phenomenon, we can amplify very, very tiny changes in the cavity. So no matter if it's a physical, chemical, or structural changes, it will be amplified. And then we collect this laser emission for biosensing and also for bioimaging. Right? So why do we need this laser-based detection compared to traditional fluorescence-based detection? So most people use fluorescence, fluorophore, to label the samples and to achieve the spectrum or the imaging. So if you compare these two, fluorescence is a linear signal, but it provides a very huge spectrum over usually 100 nanometers of wavelength. While laser is a very directional emission, it has a pump and intensity which is nonlinear, and so it has a threshold. A threshold means create like a gate to open or to turn off the signal. And the spectrum is usually very, very narrow. So you can expect a very high, strong spectrum with a very, very narrow line width. So based on these features, we have strong intensity, high controllability, and high contrast, and high signal-to-noise ratio. So all these features can be the benefits over traditional fluorescence-based method. So we take advantage of these features to do sensing and imaging. So as I mentioned, the concept of biolaser is you can replace this material with all types of biological media integrated with cavity. So the cavity can be all types of cavity. It doesn't really have to be two mirror. You can use spider, you can use ring resonator, photonic crystal, whatever else. Doesn't really matter. And then you can generate biolaser. So in the past, we have actually explored this field by using a lot of different molecules. So we use DNA, protein, enzyme, leaf, jellyfish protein, virus, and also bacteria to demonstrate lasing before. And the most fascinating part is cell laser. So we actually focus more on cell laser. So cell laser means that when you put a single cell and you sandwich inside two mirrors, and then you can expect that when the light comes on this cell, it resonates, and then it generates laser emission. So this is a laser spectrum of a lot of cells, and this is a laser image of a bunch of cells. So these image and these spectrum are very unique. So these are the information which cannot be provided by fluorescence. However, there's another type of cell laser, which is you put a droplet or a bead inside of the cell, and then this cell itself can generate laser. So this cell does not require two mirror anymore. It can itself, be flowed through the microfluid, and this cell can generate laser emission. So we take advantage of this type of, cell laser, and then we can do cell barcoding because it provides a very unique spectrum. So these spectrum can use to be identify the changes or the location of the different cells. So the basic goal in this field is to use this laser-based method to do sensing and also to do imaging. So our goal is to develop different biolaser, which can be integrated in the human body. And this is not fake, actually. One of the scientists did use some tiny laser and injected in cow eyes to pump laser emission. However, this is useless, but it's just an example of demonstrating a biolaser. So here, going into more technical parts, I'm going to show how we can use biolaser for sensing. So one example is to use a fiber and putting blood inside. And we use a FDA-approved dye to label a certain parts inside. So when this dye is binded with a certain protein or lipoprotein, it actually generates laser emission. So the mechanism works like this. If pure dye is flowing through the fluid or the ring resonator, nothing can be detected. You will see just very clear, no noise at all, background. If you integrate and flow some lipoproteins or elements inside, when they bind together, you will generate laser emission. So this can be a very, very powerful tool to identify certain molecules. Because when molecule binds together, they can generate laser. When nothing is binded, nothing is detected. So this gives you a very, very high contrast and high signal for biosensing. And we also use this method to demonstrate that actually this can be applied in whole blood. So we use whole blood to detect such molecules or proteins inside and generate laser emission inside of human whole blood. This concept can actually also move on into cells. So if you put, instead of micro size, if you put a nano size semiconductor nanowire inside of the cell. So this cell has 1 or 2 nanowire inside its body. And when the environment of the cell changes, or the inside the chemical changes, this laser will also change as well. So taking advantage of this laser that's very sensitive to environment, we can use this as a way to do sensing. So this is an example of a cell which contains like one nanowire inside. And then you can see that when you increase the surrounding solution with, for example, NaCl solution, the lasing wavelength will shift. And then it goes back and it goes forward again. So it's like a switchable laser detection inside of the cell. The next part, which I'm going to show today, is how it can be applied to imaging instead of just pure sensing. So, for example, if I have two pieces of mirror and I put cells, or if I put human tissue inside, forms like a sandwich. Actually, the real structure does look like a sandwich. So you put two mirrors and a human tissue inside, and you label a certain DNA inside of the human tissue. And then you pump this tissue, and then you will expect it to resonate, and then you will generate laser emission. So this is a traditional fluorescence microscopy, and this is laser emission microscopy. So laser emission microscopy will give you a much higher resolution in terms of spectrum and in terms of image. So these tiny dots, we call laser stars, can actually provide a much higher resolution to identify certain features compared to traditional fluorescence, like this, the cells, and this is laser. We also applied this concept to real cancer tissues. So we actually recruited a lot of patients, more than 50 cancer patients and also healthy patients. And we actually are able to identify the laser features inside of different patients. And we found that actually healthy cells has a lasing threshold which is much higher, while cancer patients has a lasing threshold which is much lower. So which means there's a cutoff between healthy and also cancer. So in this way, if I pump around here, I am able to distinguish between cancer patients and normal patients. So by using this method, I can further apply to more patients to do some data analysis. So we did a lot of image, and we also did a lot of the spectrum analysis. And then we found that actually most patients do have much lower threshold if they are cancer patients. If you are healthy, the threshold is much higher because it overgoes a regular cell cycle. So then we can use this method to develop a new type of imaging. So as you saw, it's a two-piece of mirror, and then if you scan it across the whole image, then you will be able to form a new type of a large area imaging method. We call it the laser emission microscopy. The reason why we call it laser emission microscopy because what we collect is laser. We're not collecting fluorescence emission anymore. So we can see that normal patients, usually almost nothing, while cancer patient has a lot of lasing emission. So you can see the sensitivity is about 97.5, and the ROI curve can provide an area of 0.98, which is perfectly well. So this is a basic comparison between fluorescence and laser. So if I have two fluorophore, or I have two dyes, which has very, very similar wavelength, for example, FITC and neutral, doesn't matter. So they are very similar in terms of fluorescence. If you label the same patient, you will see that the images look similar because they all present green. However, if you put this inside of a laser cavity and then you excite it to generate laser emission, you will find that actually laser emission is very unique. So these two dyes can be separated perfectly without no overlapping in terms of wavelength. And the images are actually very unique and very different. Of course, there are a lot of physical information back beyond this. However, this is just to show you that laser actually has a very unique method to provide a unique spectrum to distinguish between traditional fluorophores. And the image part can also provide very unique information other than traditional fluorescence microscopy. Next is the third application, which we would like to show, is how this laser can be used for tracking and recording. So, as I mentioned, this laser can not only be created with two mirrors, it can also be created inside the cell. So if I have a very tiny semiconductor nanodisk or 1 microdisk, which is small enough to put inside the cells, and then this nanodisk can provide 1 laser spectrum. So each cell will provide 1 unique laser spectrum, as I mentioned. So they don't overlap with each other, and they can have a very unique laser spectrum. So if I have a bunch of cells, for example, if I only have 1 cell, 2 cells, 3, or 6. So if I have a lot of cells, or if 1 cell has a lot of beads, I can actually use these spectrum very uniquely to identify where the cell is going and where the cell is circulating inside the tissue. So this is example by putting these semiconductor nanodiscs inside of mice, and then you can track in the tissue which wavelength versus the location these particles are going. And of course, this can be integrated inside the tissue. So if you have a tumor spheroid, a tumor tiny tissue, which actually grows over time, each cell has 1 semiconductor nanodisk, for example. Then you can track all these cells over several hours, and you can identify by the color, because each color is very different by wavelength. And then you can see how these cells inside a piece of tumor migrate over time by using this laser method. And then you can track it in a different speed, so that you can identify by different sizes, wavelength, and you can track these cells by low mobility or high motility. Finally, we would like to show how this can be used as a device, or how it can be used by integrating with neuron. So, for example, if I have a piece of 2 neurons and I put a neuron inside, this neuron is labeled with a certain fluorophore. And we all know that neuron has action potential. So when the neuron is excited or is not excited, it actually does have